only 2% of the VC fund goes to women. How hard it was to raise funds from 15-17 funds.
All I knew was that my idea is so strong that honestly I would invest in it.
It's important to remember for everyone, like believing in yourself, openly share the knowledge with 20,000 plus tweets.
I was at an internship and I'm from India, right? So this has all been so crazy for me doing data engineering. My boss at the internship, it was a YC company in SF, he said, listen, you're probably going to get a return offer. Don't take it. And I'm like, what do you mean?
If you're thinking of editing genes, can we like create next Ronaldo, next Messi, next Virat Kohli?
No.
No.
That's my answer. Being a founder of a company that's trying to cure cancer, sounding crazy to everyone around me.
Excellent example of measurement in physics, chemistry versus biology, bringing the measurements to biology.
I'm a computational biologist, and the amount of data that we have about the cell might sound a lot, but it's nothing to me. I'm a scientist and it's my responsibility to not say that, oh, we can go through your skin into your body, like to your kidney, and figure out whether or not you have chronic kidney disease. The laws of physics kind of get in the way.
I want to first of all visualize our viewers what is happening. Why should people care about new methods in healthcare? Why spend billions of dollars? Why do we need such technology right now? What is wrong with just putting an injection in your hand? And like, why do you need non-intrusive methods? Why do you need to split genes? So hello everyone, today I am in one of the biggest biotech labs. They are surrounded by billion-dollar funded biolabs trying to cure cancer, trying to discover diseases, lot of intersections with AI, neural nets, and most importantly, the future of cancer treatment. We're going to get to know with Parmita and Ahmed, CEO and CPO of Precigenetics, which is a next level startup where you could study the DNA, study diseases without even injection, which is known as non-intrusive methods. You can get DNA out of it, discover so many new things, and cure cancer, prevent diseases. So much to learn. So can you please introduce yourself?
Yeah, for sure. Thank you so much for this opportunity. So my name is Parmita, and I'm from Delhi. And my parents are doctors, so I've always been really interested in how science could help people, like, you know, basically out of impossible situations. And that's why I came to America. I went to UPenn. I studied computational biology and computer science at Penn, and I was also researching at Penn Medicine. And that's when I decided to start Precigenetics, and we recently moved the labs to San Carlos, California.
Oh, very nice.
Yeah.
Sure. My name is Ahmed. I'm a CPO, and my background is in computer engineering, and everything I do is to remove barriers and enable others. So I used to build tech for the blind, and I'm very excited to have joined Precigenetics to remove barriers in drug discovery and in the future in the clinical setting.
I got to know about you through Twitter. You are really active. Have created a founder-led community. Okay, you have engaged with Elon Musk too.
Okay. Yeah, I think that Twitter changed my life. To be honest, it was actually so positive because it was a part of why I was able to start the company. Any other time it would be considered insane to start such a company, but I had reached a point where the people around me on Twitter, they know me and they understand me. So how did I get there? I was all alone. I'm also from, India, and like I was studying a lot, but I was just going through a lot in 2023, health-wise and so on and so forth. So I would do my work, but instead of, doing other extracurriculars in college and stuff like that, I just wanted to post about biology. And why? I just had this weird realization that engineers don't understand biology enough. And I'm an engineer and a biologist. So you know what? I want to just teach people how they can help biology, right? So one of the tweets I'd posted was if I had $1 billion, then I would, make this microscope that can tell cell states non-invasively over time. I kept saying this kind of stuff for like a year. I'm full of ideas. Like I have ADHD. I'm full of ideas. And I just saw no one was acting on it. And I realized that maybe that's because I should be acting on it. So really, like, it all started as a super informational thing. And I was going to, be into academia, like, the PhD program, this and that, and I do all these things. But I realized that most people do not want to communicate this the way like I do, like how insanely I feel about this. So I ended up meeting some of the best investors in the entire Silicon Valley through Twitter.
Wow.
And it's just because they like the information and they seem to believe I have a good taste in biology. Like, nice. And so, yeah, I realized that, okay, if there's anyone who should start this company, it's me, because I came up with the idea, right? So yeah, like it was all connected to each other. The way that I tweet a lot is instead of using my notes app, I just like tweet the things that I usually, or I would spam group chats and my friends would get very annoyed with me. But I was like, you know what? This is so cool because you can just post stuff and it's nerdy. So you're learning something, but no one is annoyed in a group chat. So it's great.
Totally. Only 2% of the VC fund goes to women. How hard it was to raise funds from 15, 17 fund.
The craziest thing is that this is very true. And I'm so glad I didn't know that number before I went to raise, right? I think that it's actually the easiest thing to raise from people who actually want to invest in you because they believe in you, right? Mm-hmm. So all I knew was that my idea is so strong that honestly, I would invest in it. That is how I went into those meetings. Especially with 1517 Fund. It's one of the most successful funds at the pre-seed in the world, right? My investor, Michael Gibson's written a book where he talks about this. Their biggest sort of thesis is that if you are leaving everything in your life, including like traditional accomplishment, which I would get if I'm at a prestigious job, the offers I didn't take and stuff, to do something sci-fi and compelling, then that is an extreme green signal that you're someone to invest in. So above anything, I want to be grateful for not just 1517 Fund, but Jim O'Shaughnessy at O'Shaughnessy Ventures, Chaotic Capital, all of our investors for believing in the idea before there was a citation that I could give you, right? Because that's the crazy thing about investing as compared to science is, no, you have to give the vision and then the evidence comes in. So now we have the evidence, but back then you just went by what you felt compelled by. So thank you so much to my investors.
Excellent. I think it's important to remember for everyone, like believing in yourself, and you not just believe in yourself, you openly share the knowledge with 20,000 plus tweets. That's not easy.
And 25,000 people. Yeah.
Yeah. Like it's not easy. Like giving that much knowledge, what I've seen on your Twitter page, it's incredible.
Thank you. It's been a lot less recently because I've been using it to do things. But when I was a student, and this is my advice to all students, like everyone told me you're going to get more money as a Facebook software engineer, data scientist. And that's why they wanted me to do comp bio because, oh, you know what? Like you are more mathematical in how you think, but you like bio, but okay, fine. You just do the bio, but you'll actually end up at Meta, right? Right. And I'm like, no, I don't know, right? Like it just doesn't sound right. Mm-hmm. I was at an internship and I'm from India, right? So this has all been so crazy for me doing data engineering. And my boss at the internship, it was a YC company in SF, he said, listen, you're probably going to get a return offer. Don't take it. And I'm like, what do you mean? He's like, don't take it if you are truthful about how you talk about biology, because I think you're meant for something else. And multiple interviews that I would have for data science, by the end, I would be told, I don't care if you get this job. I've never seen someone this passionate about the field. So I'm like, okay. And yeah, it just kind of, that it just worked out. It was the right bet.
I'm going to repeat. It's important to remember less than 2% of the VC fund goes to women. Out of that, less percentage goes into specially like computational biology, and even less goes into someone under 25, and even less someone who's an immigrant.
Really commendable thing.
Okay, that's really commendable. So you tweeted that viruses are not alive.
Yes.
How did that story go?
I tweeted that viruses are not alive, changed my mind. And at that point, I'd been tweeting a lot of biology, but I don't usually have opinions. It's all science for me. So this one guy got very offended. He was like, "You're not a professor. You're just a kid. Oh, you don't know anything. Why do you act like an expert?" And I'm like, dude, I'm a researcher. I never, and any person who knows anything about the world would understand why viruses are not alive. They have metabolism. But this guy, like, he just tweeted that, and everyone was just like, all the sexist people were just kind of like talking in the comments. But for some reason, like a huge page, which I will not mention, unintentionally retweeted that. And then all of a sudden, like, I was still a smaller account, mostly known amongst like, bio people or whatever. And everyone's like, and my professors, my own professors, like the best professors in biology are defending me. She hasn't said anything wrong. She doesn't say anything inaccurate. She's like very, and literally that's the biggest cancellation I've ever had.
Really?
Yeah. And it was fine. Like, it was the first one's always very difficult because you see all these like very, very sexist, racist people. But then after that, I've just not cared. Like, and I just don't, I'm not problematic. I either appreciate someone or I don't talk about them. Like, that's it. So literally you just have to go through it once in your life. And a lot of people told me then, you grow thick skin. And that's been the most freeing thing in the world to me. Because if that's the worst thing you can come up with when it comes to me, then I must be doing something right. Like
100%. What doesn't kill you makes you stronger.
But that happened when I was looking for jobs. So can you imagine? I was looking for jobs, and then guess what? I was like, I'm going to give myself a job. It's okay.
Oh, nice. And you found it.
I was like, who cares?
Amazing. And I want to, first of all, visualize our viewers what is happening. Why should people care about new methods in healthcare? Why spend billions of dollars? Why put millions of dollars? Why do we need such technology right now? What is wrong with just putting an injection in your hand? And like, why do you need non-intrusive methods? Why do you need to split genes? Let's start with the background.
Yeah, for sure. So I think that, I came up with this idea when I was doing research, and really luckily Penn has a really great biology department, and I was working in single cell epigenetics, and I realized that we use sequencing for everything. And the problem with sequencing is that you have to kill the cell to measure it. So that sounds completely fine, right? Okay, yeah, you're destroying a sample, but you're getting the readout. But the problem is that past DNA, everything's dynamic. So like, for instance, just to draw a parallel, this is not what we're doing, but if you have like a once a month glucose test as a diabetic, as compared to having it continuously every single day, what is the difference it makes? Well, it allows you to know what food is causing a glucose spike. It allows you to act proactively, but also it allows you to understand your condition, right? Mm-hmm. Now, this happens in the morning when I'm stressed out, this happens, and so on and so forth. So now, when it comes to cells, these are very important for understanding diseases, because when it comes to making drugs for diseases, therapies for diseases, you need to understand, you can't just like keep on giving drugs to, mice and basically, this works for this one mouse, so and this is an oversimplified representation of drug discovery right now, but a lot of it has to do with, basically relying on data that's not simulating real life. In real life, unlike chemistry, where, you can take a sample and destroy it and you know what it's made of, in biology, it's like always moving. So as a result of that, I realized that we need to use better optics. Every great invention in biology usually comes with a measurement technique. If you look at the first microscope, changed the world. Eventually, a microscope allowed us to figure out that cholera is like related to microbes and not like, it's like the transmission of cholera is like through water, for instance, right?
Cholera?
Yeah, just it's this disease.
Cholera?
It's a disease that basically confused a lot of people. Back in the day, there was a doctor called Jon Snow, not from Game of Thrones, who basically was able to deduce that people thought that all kinds of things like, oh, it's because if someone smells bad, then that they can give you cholera and all these crazy things. And then Jon Snow basically figured out, through the water canal system that, oh, the incidence is like more in these areas, so that means that it's related to the water. And then they stopped the flow of water, and then it actually helped change the situation and stop the epidemic of cholera. And then at the same time, there was a microbiologist, I believe, who was able to figure out through a microscope and sort of like confirm that there is this pathogen that's like causing cholera. And without that, we wouldn't have like 100% known. Eventually that led to us being able to, for instance, use an antibiotic, right? Against like how would we have known? Sure, we know that it's like through water, but what exactly is it in water? These things you figure out through microscopes. Another example, sequencing, right? Sequencing has told us so much about the human body, and now we're at a point where we can use CRISPR, and we have entered a new generation of genomics. So we're looking for the next big measurement, and what I realized is that it's not going to be sequencing. It's not the same thing again. It never is, right? And so it turns out that I was looking at, trying to infer dynamics for single cells. And I'm a very computational, so I work with huge sets of data, right? So I was looking at these sort of dynamics, and I was like, wait, I don't have any data in real time. So what if I had a camera that could just like capture in real time without harming the cell what is happening? How would that change things? And that's when I kind of realized that this is the future. And if you combine this, the problem with this is that there's too much data that you get. But if you combine it with machine learning, then actually that's enough data for machine learning to infer dynamics.
Yeah, another example is what you gave me with chemotherapy. You kill some cells, but non-targeted cells also die in the process. That's why you get side effects.
Yeah, I was mentioning, and you can touch on this, was how, if you just go back to what you were talking about, in drug discovery and in a clinical setting, one of the major problems is, as Parm said, biology is currently stuck in these static snapshots over time. But biology is dynamic. All of the cellular interactions or the protein-protein interactions and so on happens in this dynamic timescale between these two snapshots. So what we're trying to introduce is we're trying to bring FaceTime to the lab as opposed to Polaroid snapshots. And we're giving that window to both researchers, scientists, as well as to machine learning in order to decode these interactions. And like, as Parm, you can mention how like 97% of drugs fail
Cancer drugs, yeah.
in drug discovery. And a big percentage of this could be due to the fact that we don't have better measurement tools. We need better measurement in order to inform the insights and in order to inform everything that comes beyond that. Now, when it comes to what we're talking about, maybe we could talk about that after we talk about like how do you find targets, how do you build the drug in order to bind to that target, and so on.
Totally. So, like, there are so many applications connected, as you mentioned. When you can precisely see what's going on and understand even better, you can build better stuff out of it. For example, people are creating pancreas these days. It's all happening because of precision. People are creating new organs. They're planning to create them. There's so many applications. But can you once again imagine or like help people visualize a future with a startup like Precigenetics? I want to make people visualize.
Yes. So I think that, it is very easy for me to like go ahead and overpromise and say, we're building the Star Trek tricorder. Everyone imagines that the second we like bring this up. My problem with that is I'm a scientist and it's my responsibility to not say that, oh, we can go through your skin into your body, like to your kidney, and figure out whether or not you have chronic kidney disease. I'm sorry, but the laws of physics kind of get in the way. But at the same time, there is a lot of very real alpha here, which comes from the fact that, firstly, you start with a more boring reality where instead of doing like just thousands and millions every week of like, these screening interactions to try to figure out, using these snapshots, whether this drug is interacting with the cell correctly, and therefore should it be sent to the next stage of, let's say your clinical trials and so on and so forth. That would stop a lot quicker if you can actually you're not just playing this probability game. Oh, did I catch it in time? Oh, did it? Or, there's a lot of things that are we don't know the time scale of in biology. So that's the biggest thing, where you're basically going to be able to look at the same cell across time. And it's kind of like you actually understand the biology of what's happening. And the reason why this is so shocking, for others it might sound boring, but actually you have to look at the state of the art in biology. We don't know the mechanisms of action for a lot of the drugs that we take into the body. In fact, for instance, with retinol or something, we use those medications for, say, acne, and then they discovered in the process of development that it's causing anti-aging. And that's how we figured out minoxidil was the same thing. It's like, wow, it's causing you to grow hair. You didn't know that. It wasn't a targeted approach. And so a big part of that is the data, the ability for a computer to handle the data, which we have finally reached that point with, all these changes in AI and compute as well as a big one. But the other part of it is being able to give the data. So that's one thing I wanted to say there. Now, when it comes to imagining a future, this is where I will go into, like, imagine. Right now we're doing drug discovery using this kind of a microscope, what we're calling a microscope. But it's a non-invasive microscope. With sequencing, you're destroying the cell. It's different. You cannot go inside the body and use the sequencer because you'll be destroying your cells in the process. You can't just, remove all of your body and then like sequence it and then bring it back in. That's never going to happen. But because this is a camera that's like this camera, hyperspectral camera, so to speak, where you have all of this information. Why not? There's no reason by first principles why you wouldn't bring that to this operating theater. So imagine like someone has brain cancer. That's something we're looking at. You have to figure out what cell has the cancer, what cell doesn't have the cancer. So the surgeon, and my dad's a surgeon, so this is very exciting for me. You can go in with a probe and basically figure out if something's a cancer or not a cancer, and then remove it. And how you're doing it is you're using the machine learning assistance, right? That comes from interpreting this data, right? And then you not only are able to understand that, but then based on that, you give the right treatment. And that is very important, because till now, we have been giving treatments we don't have personalized medicine, right? When it comes to cancers, you need cocktails of medicine. So you're increasing the chances of, someone surviving. And then eventually, you can go into a clinic with similar probes for melanoma, right? Which is like at the skin level, or even hopefully at the blood level. One day we can crack that, sort of limit. But, it's going to take some more capital to get there. So in short, the way I would put it is we start with the drugs, right? We start with the drugs, and then we want to move into using the same devices that we use for drug discovery in trials, as well as eventually in diagnostics, right? And so now then the diagnostics, like to the extent that HIPAA allows, right, will be informing the drugs, and the drugs will be informing the diagnostics. It's like a loop.
Okay, so it's a complete circle.
Yeah.
You understand the drug in depth, you understand the organs in depth, you understand the cells in depth, and then it goes back and forth.
Right.
That's what you're saying. That's a complete circle of path. And directly making a conclusion, like, a good example could be of Elizabeth Holmes. She just gave a really big vision, right? That's how it failed, and overpromised and gave fake results, and then shocked the world.
If I'm being honest, I think that Elizabeth Holmes was, she was after something crazy, and it involved microfluidics. And to some degree, what she's doing has somewhat been realized with some companies recently. There's one out of Stanford, and then I think there's one in Canada called Vital Bio. And the only difference is they're doing it realistically. So the biggest problem with Elizabeth Holmes was that she was lying. It was not at all with the fact that she had a dream. And if she went about, see, I also wanted to start with like a Star Trek tricorder, right? I was excited, but then I realized, you know what? I need to be able to achieve something that's foundationally in science, right? Like you cannot just make things up in science. That's the difference between science and engineering. It annoys like a lot of people who are engineers. Like even in our team, like sometimes they're like, no, just tell us exactly what it is that I'm like, no. This is an iterative process. You can never know until you've experimented. So there's also a bunch of other differences, right? Where it's like she went to like a CVS and like all this stuff, but like, did she go through the FD? I don't think she had data to show for. And the biggest blame I would put on like, I don't know, all these media people who just believed everything she was saying. And then now people just don't believe biologists at all, which is like so crazy to me. Yeah.
So the struggle is harder after Elizabeth Holmes' scam on Theranos. So you think it was harder for you to raise funding, harder to convince investors?
I think that if I'm honest, like it was more of something that brought anger to me because we're not like making some optional product like, oh yeah, maybe we will cure cancer today. Like no. And I'm not talking about just myself. I didn't have any such problems because the people that I spoke to know about my credibility. And frankly, like, you know, I think it's very easy not to be Elizabeth Holmes because she just lied and you just don't lie. And that's how you do science, right? So the most important thing is I am super passionate about science. I always have been since I was a kid. And that's like, so there's no equivalence there. Mm-hmm. But with that said, I do think that, you know, it's like, kind of ridiculous that for a few years, like because it's been enough time now, right? But for a few years after all that happened, we did not do much diagnostics. All the biggest harm to society, in my opinion, was the companies that were honest that had to fail as a result of a lie that they were not involved in. Because this is not like some optional industry. This is critical. And early diagnostics, for instance, are the easiest way to save people from breast cancer, from pretty much any cancer that has any treatment. So, yeah.
And regularly analyzing it over a period of time, studying the cells over the period of time. I saw the machine, how you take pictures and how precisely you're capturing the cells and like without intrusive methods, without injection, you're able to like discover over the period of time how cells evolve. So can you elaborate on what is epigenetics, what is genetics, how do you study them?
So I think especially when it comes to the brain, this is such an interesting question. And firstly, I you know, actually talked about this recently. I write a lot and I said, you know, the only thing that makes biology make sense to me is to see it as quanta. It's to see it as units, right, that are discretized and they're cells, right?
Like numbers. You just want to see them as numbers.
Yeah, exactly. It's like, like a brain, it's cells, right? Like anything you look at, it's cells. And that's the only regularity to something, metabolism, cells, like a couple of other things that will exist across life. So I love that about, you know, biology. And so from that perspective, when you're talking about epigenetics, it's really important to remember that all this time people have been telling you that your DNA, your genes, they code for your body, right? Mm-hmm. So every single cell in your body, that means has the exact same genes, somatic cells, right? They all have the exact same genes. So my question to you is, how do you have skin cells that look like skin cells, and then you have your brain cells that look like brain cells? Like you've seen brain cells, right? Like with the axon and everything. But that's so different from like a skin cell.
They have still same genes.
Exactly the same genes.
You can discover genes, DNA from hair, from saliva, or anywhere.
Yeah. And
same DNA.
That's why you can, because you can use your saliva and you can understand all of your DNA, right? So how is it possible that you have the same code, right? But it's giving different outputs? That's a question for you. What do you think?
Same code, different outputs. So you're saying cells evolve, same code, DNA is same, but cells evolve and gives different outputs over a period of time.
Different shapes, different functions, different structure.
Reactions with the environment, it will react differently here, different in space.
And also, some of them will divide a lot, some of them will not divide much, right? Your terminal neurons, they can up to theoretical limit of 200 years, they can be alive, right? But then some of your neural stem cells, they're going to keep dividing before that. So the point is that all of the cells have the same DNA, but the thing that governs how that DNA is expressed is epigenetics. The epigenetics is really anything that is, expressed that is not explained by the DNA, you could say. There's different definitions of epigenetics, right? So this is really important because this is the mind-blowing thing is, everyone's been telling you your genes are your destiny, but even within your own body, you can point at two parts of your body and see completely different cells. They have different lifespans. They have different. So this all happens as a result of cell types. And for the longest time, we actually assume that cell types are these discrete things. You have a certain kind of a progenitor cell, which is an intermediate cell between a stem cell and a neuron. It's just, it's complicated, but basically you have a stem cell, then you have a neuron, and then how does a stem cell divide into becoming a neuron? It goes through these phases. But what we realize is actually, no, this is a continuum, right? And cancer is one of these sort of cell states where, your genes, especially in some cancers that are epigenetic driven, they will have the same DNA. There's no mutations, but they are expressing cells in a different way. So kind of like your genes are a piano, right? Mm-hmm. And your cell is a song. So your DNA, every gene is a different key on a piano, right? But the song that's being made is your cell. So some things are on, some genes are on, some genes are off, right?
So different algorithm making a cell, making it different, but the algorithm is same all over, same piano all over, same DNA all over, same genes all over, but with the combinations it making different cells.
Yeah.
That's why cell over a period of time is different. But at the end of the day, DNA, genes, which makes you unique is same.
It's kind of like saying the API is the same, but the function calls are different in different cells. Does that make sense? It's kind of like, you're calling, you have the same exact functions, right? All of them are the same. So one of the functions might be, and this is not actually a gene, but let's just say, you have to live for 200 years as this cell. So then you call that, and you don't divide into other cells. So those two functions, you're going to call it in the neuron, but you're not going to call it in a stem cell, for instance.
I'm going to call it the neuron, but going to call it the stem cell.
Oh, you're going to call it in the neuron. Okay, maybe I confuse everyone.
And one other interesting.
Wait. I didn't understand.
Yeah,
I'm gonna call.
Not gonna call.
I got lost.
Okay. Let me make it a little bit simpler, and this might have to be cut here.
Example, I don't think I can understand. I can think about can you explain in cooking form, same ingredients, different dish. How about that example?
Okay, fair enough. But it's in a kitchen, it's if you take salt, then you have less salt. You don't have that problem here. So how I'll say it is, if you have keys in a piano, but you're making a song, you're not going to use all the keys, right?
Mm-hmm. Some will be on and off.
Yeah, some will be
neurons. Yeah, on and off.
In neurons, there might be some keys that are on, some keys that are off, right? And that gives it the chemicals that you find in the neuron and the structure, right? But in other cells, it's a completely different song. It's a much lower song. It's using a different range of keys.
Yep.
So what I'm trying to say is it's kind of it's not just your genes, it's how your cell is reading them and expressing them. How many times is it expressing them? And that is very important because it adds a level of almost agency, right, to life as a whole, because it's like you're not just, it's not all deterministic. It's not like, oh, you have this gene, that means that you're going to have a certain fate. It's like, even at the cellular level, it's like your fate is, dependent on what you express, what you don't. If you have a book in front of you and there's one chapter that talks about how to say curse words, right? But then you staple that chapter, then you're never going to learn the curse words, right? Because yeah.
You're closing you're not going deeper. You're just
you're just
hiding
not
things
reading it
in the house and locking it.
Exactly.
I see. So
that's epigenetics. Basically, epigenetics is the reading of your genes to create these different, outcomes across different cells. Now, this is one thing that's important here is if a cell, it looks like a neuron, or kind of looks like a neuron. It's like in between a neuron and a stem cell. And it has and it divides into another neuron. Mm-hmm. They both are expressing genes in the same way, right? Because your organs, they have cells that all are very similar to each other. Mm-hmm. Right.
So can we say in similar way, it's like basically neural network is a black box for humans, for even the AI models, or even machine learning. Now you're saying that we are discovering that black box, discovering what's happening, how the algorithms are going behind the scenes, how neurons are reacting, how cells are reacting to finally give you the song. We are setting the song and we know the piano, but we're setting the song.
So it's kind of like if this neural network is your DNA and like your neural network or your DNA is different than mine, then in one snapshot of time, the weights that are the epigenetics or the weights on this neural network is kind of the expression level on each node of this gene or node in the neural network. So it's kind of as if all current methods that are able to understand or extract DNA or sequencing, they're able to see the nodes in the neural network, but with epigenetics, we're trying to see the weights in these nodes.
I see. So if a DNA sequences, if we see, if we can see the sequences, but we cannot see the weights deeper down it, now we can see the weights, we can manipulate it, and by manipulating it, you can discover so many new things, pancreas and all that, right?
Yeah, I think that the short of it to me really is that these weights, they change over time. So the short of it really is that you have a dynamic aspect to life. If you look at the sequence, my saliva sample, and you cannot figure out, you might be able to tell, oh, I am more likely to develop this disease or that disease, but you cannot tell, oh, by this age, you're going to develop this disease, right? Or, oh, you're going to have this disease at age 20, then you're going to have this disease at age 25. That's impossible. Why? Because your fate is not decided the day you're born, unlike what a lot of Vedic astrologers say, right? It's not, you have a dynamic life ahead of you, right? So, for instance, you eat certain things and then it can cause certain responses of changes in expression, right? So when I first got into this field, I was thinking in that way. Oh, wow, epigenetics means if I eat haldi, then I'm gonna not get cancer. That's really cool. But then I realized that that's what I want, but that's not what it actually is. True that curcumin, the active compound in haldi, is one of the most well-known epigenetic modulators to protect against various diseases. Mm-hmm. But you don't know the mechanism at a cellular level, right? That's where a drug would go. Uh-huh. If you were to give a drug, if you were to give a high concentration of, say, curcumin, for instance, you need to know where all it goes, what all it causes, whether or not it always works. And so, that's when I came across single cell epigenetics. And single cell epigenetics is where you're basically studying, instead of how a human being is affected by certain food, you're studying how a cell is affected by certain environments and so on and so forth.
100%. And it goes beyond by splitting genes, splitting cells as well. Can you elaborate on that? Why should people care about splitting genes, splitting cells?
I wouldn't say we're working on gene editing. So that's for instance, CRISPR is gene editing, right? Uh-huh. You're editing a gene, right? You're adding a new gene. You might use a guide RNA. You might, cut your gene inside your cell in a specific area, remove a gene. Let's say you have sickle cell anemia. you remove that gene and then you add, like it's actually not, you're removing a specific portion of your DNA and then you're replacing it with another portion of DNA that you've synthesized, presumably. So that's CRISPR. With CRISPR, some problems are with translational, the translation of CRISPR. Obviously very expensive, regulatory, market risk, all these other things. But the most important thing is that if CRISPR could have cured people who are going to die in 18 months of brain cancer, right, then they would have used it. Because these are terminal patients, like, there would have been trials for such a thing. Unfortunately, it's not that simple in most diseases that affect most people because they're not, like sickle cell anemia is a genetic disease. You have it from the day you're born. If you have those two recessive, like you have it from the day you're born, right? But most diseases come, like they come as you age. They have an onset. You take too much of something, you do something, and then that disease happens. Autoimmune disorders, right? So that's what is affecting so many people. So we understood CRISPR. We figured out CRISPR. And then we figured out sequencing, right? So sequencing is the reader there, right? And then the CRISPR is the writer. It's like being able to make changes based on what you know from sequencing, right? From sequencing, you know the genes. So you look at in sequencing sickle gene, sickle DNA and then normal DNA, you can tell what's different, right?
Okay, with sequences you study the genes, and with CRISPR you can fix the abnormalities, and after fixing that, maybe even cure the cancer. But it doesn't work out. Why?
It doesn't work out because we even know, like, P53 is this one specific locus where it's just P53. That's just like a gene that we know there is a change in that specific locus for so many cancers. But the problem is that's not where it starts. Like,
you need to study the timeline.
Exactly. So this is the kind of thing it takes a little bit to like start getting it, but then once people do, it like makes sense. But really, like, unfortunately, most genes are not only responsible for giving someone cancer. If you have autoimmune genes and you remove all of your immunity genes, then you're not going to be able to survive any disease, right? Wow. So that's the biggest problem with cancer is that you cannot just, it's not a binary thing. If you completely remove every gene, like encodes for a protein, which can be life-saving for a cell, for an organ, for you. So yeah, you cannot just completely turn it off. Not just that, but also you have trouble like targeting the right cells. So for instance, with the brain, how do you get inside the brain? There's a barrier that prevents you from, most things cannot enter the brain. So those are huge problems. So even though CRISPR is amazing in so many ways, and I think it's truly tremendous, I don't think that editing genes, is a solution for the most difficult diseases we deal with today.
Okay, so I think you guys, by studying the timeline, having the whole story of a disease, for example, HIV hits your immunity, and immunity over, you're dead. So like, you won't die because of HIV. You die because your immunity is so low. Any other disease happens, you're over.
Exactly.
So that's why, like, if you tackle that immunity, you solve it by editing the genes, and you mess up with immunity, you're gone.
Exactly.
Yeah.
And that's why, by the way, this was a meme I was involved in on Twitter, but that's why I have a huge problem with the whole meme about that guy from, like, Dr. Jian Kui He, from China, who basically posts pictures of himself, like, like a model in the lab, right? And then writes stuff like, oh, we're going to edit the genes of embryos embryos don't even have a choice. And also that's okay if, like, it's a life-threatening condition, but you don't know what, like, you're not talking about, like, with sickle cell disease, you don't need to do it at the embryonic stage, right? But when you're doing it at the embryonic stage, you don't know what will happen. You've only ever tried this on a mouse. So what will, and then that edit, the child of the future human, the embryo, is going to have that edit. You're always, it's going to pass along generations, right? And so that's why, like, one of the things that happened on Twitter was I started this kind of a fake funny beef with this person, because I was like, why are you all laughing at this? This is ridiculous. So that's it. I understand the need for gene editing for a lot of things. All I'm saying is it cannot tackle everything.
Analogy here is with X-rays, you can analyze bones, but you cannot analyze blood. For blood, you just have to take out with intrusive method. So you are expert in monitoring over a period of time. What do you see the future here?
So this is the cool thing about our technology, right? Whenever, usually, so as I said before, we're starting in the lab. I mean, you could come to me and say, hey, like, if you can look at a cell, that's fine. We have had microscopes. How will you use that through the skin in a person, right? Like it's not that easy, right? To just surpass the skin and like look at the blood. Mm-hmm. And I agree. It's not easy because it's not standardized. It's not easy to calibrate. People have different skin colors. People have, and then you have lasers involved. So then there's a lot of regulatory burden, but it is possible. And that's been known, and that's been done in mice, and so on and so forth. Mm-hmm. So initially we wanted to actually go about it the opposite way, which was, we should go full diagnostics, right? And then eventually we understand these diseases, and then we can go into the actual therapeutics. So how we do that is we look at drugs, and then we look at how they interact with cells, so that the same drugs that 97% of which would have failed in trials for cancer, you're looking at it and like maybe looking at some drugs that other people are not looking at, using AI techniques and so on and so forth. And then you're going into the lab. Like, we're not doing this all like, on the computer. And then you are able to get very high dimensional data, right? And then use that to figure out what drugs are, going to be likely succeed in the trials. However, with diagnostics, the problem is that if you start with diagnostics, you cannot use that data for drug discovery, because it's like there's HIPAA compliance, there's ethical reasons, it's just becomes a slippery slope. Not just that, but you have regulations to deal with.
Yeah.
Like FDA regulations for the device itself, right? Because like it's laser, so you have to do that. And what we realized is that actually, if we focus on the drug discovery first, so we start at the cell level, then we will become so good at understanding very fast what's happening in a cell that we can then like sort of multiply that across the cells in a tissue, and then eventually move towards actual diagnostics in human beings.
Excellent. So the example you said is with mouse, we can study the cell, we can take it out with a laser, but with humans, you cannot. It's not apples to apples comparison, it's apples to oranges.
Yeah.
So you cannot just like put laser in human being. So you have to study with imagery, non-intrusive methods, study the cell, and with imagery, with technology, with high definition image, which you have like GBs, terabytes of data, which you compile. With AI, big data can be handled. And by handling that data, studying it over a period of time with weights, with neural networks, similarities, you study it, give the results, and then go with the flow.
Exactly. So if you know how to a disease well enough to be able to maybe not directly push drugs through the pipeline, maybe work with some companies or work by yourself. If you're able to basically understand a disease enough to understand its kind of math, to like be able to come up with the right therapy for it, then you understand it enough to diagnose it. You might understand it even better than you need to. So that's actually what precision medicine means. Like, the term when it was invented, or at least when it was taught to me, we were told there's a detect side, right? And then there's a treat side, where the detection is going to inform the treatment. I can give an example. One big thing that made me want to start this company is my friend's mom died of ALS, and so did my own aunt. She was my dad's brother's wife, and Yeah, it's amyloid lateral sclerosis. Yeah. Yeah.
Okay.
So basically, it's a disease that's like rare. Often it could be because of genes, or it could be because of some exposure to some factory chemicals or whatever. And then it causes you to lose. It's a little bit like dementia in some ways, except you have 5 years to live or something like that. Just like Stephen Hawking had ALS, right? So the problem with the ALS is that, unfortunately, like when my friend's mom had it, it was in India. And that was the second person I knew at that point who had ALS, right? And I started crying. I spent 2 days in deep depression for one simple reason. Wherever we don't know how to treat something, like there's usually something we're not measuring. And in ALS, it is rare. And I realized that if they just included, for instance, Indian patients, huge country, in, I don't know, trials or understanding in research and understanding what's happening in ALS, trying out drugs, maybe we would have more idea about what's happening, right? And she unfortunately passed away, my friend's mom. And yeah, I mean, it's okay. It really hurts because, you know, weirdly enough, the best answer, I just mentioned Haldi, right? The best answer that even the Cleveland Clinic had for her was, I guess Haldi, like turmeric, has been shown to be neuroprotective. It can like reduce, you know, how fast the disease progresses.
Really?
And
It's a fact.
well, we have such little drugs that the smallest correlations in nutritional supplements is all you can give these patients because there's like not many of them. What that made me realize is that you don't always need more and more and more and more and more patients. You don't need more and more and more and more data with the same measurement. You actually need to measure the few patients a lot better, right? To be able to actually understand. And with ALS, they came out with a drug, but it only works on 10% of patients who have one specific gene. Okay, only on those patients, like one allele of a gene, that is the only patient that it works on. So that's because sequencing already exists as a measurement technology. Now, imagine going 10 years ahead and you have something way better than sequencing that's able to tell you so much more. Than more drugs will pass trials because you're saying, oh, it's only this kind of drug that's positive for this detection mechanism that's going to, you know, that it's going to work for.
Basically saying this algorithm works for this problem, so we're going to use this algorithm and solve it because we have seen that pattern.
We're trying to make biology as readable as possible, and in essence, that will build a platform, but also enable future progression, whether it be in research, whether it be in drug discovery, and hopefully in the future in the clinic.
An excellent example of measurement in physics, chemistry versus biology, bringing the measurements to biology.
Even in like physics and chemistry, one important thing to remember is that the reason why you can measure is because you're measuring distance for one thing, you're measuring time for one thing. In biology, the most important facet of this that I really need to touch on to really make it make sense, because one of the questions that I get a lot is, why has no one done this before?
Yeah.
This is so incredibly simple. I don't understand. Parmita, can you explain? I can explain. I'm a computational biologist, and the amount of data that we have about the cell might sound a lot, but it's nothing to me. And that's important to know, because guess what? Instead of measuring distance and time and, you know, trying to solve one equation for like a couple of objects or whatever, you're just doing this for thousands and thousands and 10,000 genes in parallel, right? You're like, that's just the genes, right? Then there's your lipids, your morphology, your shapes, structures, time dynamics, right? So even being able to get this amount of data for one time point is kind of difficult. The closest we've gotten is sequencing. However, that's only for one time point, right? The problem is that we are measuring so many things that we could not get there without a computer. That's very, very good. That's important because if you notice there's a book called Codebreaker by Walter Isaacson, right? He talks about how all of the sort of like revolutions in computing and the revolutions in biology, they always go hand in hand. Always. From Fortran, they've been going hand in hand because biology is too much information.
I want to give an example. Like, everyone is trying to build something in B2B SaaS because it has more profit guarantee, higher success rates, less people building in biology, lower success rates, but the impact is really high.
Yeah.
But now computation coming up, passionate computational biologists coming up like you all, it is catching up.
Yeah, I certainly think so. And I think that when I was a little younger, because I became a computational biologist. So Emma's like from a more sort of EE and a computer engineering background himself. So I'm actually like more of like the biologist on the team. And like when I was studying comp bio, it was before OpenAI's ChatGPT. It was before COVID. And people were so confused. Like, what are you saying? You're studying computational bio. That doesn't make any sense. And I'm like, no. It does make sense. And then over time, now what happened in COVID? All the labs shut down. And then everyone was like, oh, we have all this information, so we all have to become computational biologists, right? Then AlphaFold happened. AlphaFold 2, I think, won the Nobel Prize.
Correct.
Right? So now it's like, all up and going. But I remember my mom was telling me that, hey, like, you don't know what'll happen tomorrow. And this field will become important before the pandemic, which obviously comp bio helped, a lot with COVID, right? But she said that and I was like, yeah, okay, fine. I'll wait till I'm 40 years old. At least I'm studying something that I like with comp bio, and then suddenly the next year COVID happened, right? And then all this stuff happened. So I think we often forget that AI was a niche field, right? Wasn't it? Even in tech, I was doing these data science jobs and you would say AI, people would think, oh, robots,
Yeah. Data science is now a dedicated major in so many.
Yeah.
Like generative AI is a dedicated major. Right?
Really?
You posted about UC Berkeley, right? Like data science is most
yeah.
popular major at UC Berkeley now.
Yeah.
It's fascinating. Like people seeing that coding jobs are dying. So data science major is the biggest hype.
I think this is so wrong. Like, your data science is all about domain. I was sort of a TA for a lot of like data science classes and stuff like that. And this is what my professors always told me to tell people. It's all about domain, right? It's all about domain. You cannot like just keep switching domains and like, be as invested. Yeah.
It's like chase the skills because coding is still the core skill used in data science. Chase that rather than the hype. Chase the basics. People have to perfect that.
Well, I'm not very good as a software engineer, by the way. I wouldn't say so. Like I would sleep while trying to solve lead code questions in software engineering because I'm like, why can I just not look at the answer and then just do that?
Yeah, it's about like, like things computers are good at. Why do it? We don't calculate in our head 3 to the power 256.
Unless you went to school in India, I guess, right?
Like you tweeted about if I had $1 billion, I would build in invasive biotech devices, right? So I want to understand how does research work when you have unlimited funding versus limited funding. DeepSeek is an example of with limited resources, you hack into bringing something extraordinary. It's also called the Chinese movement, by the way, and they have not allowed the researchers to come out of China. Like it's that big. And on the other hand, like with unlimited funding, the example is like, the scaling laws, data sets becoming really big, LLMs becoming really big. So what is the research mindset with unlimited funding versus limited funding?
I took it for granted how expensive the samples were that we were working with in the lab. I never thought about it when I was in university. I just thought this is all free. It just falls from the trees and, we're all good, right? These are cell samples. They all exist. They must be free. No, they're $5,000. So, I mean,
Really?
it's like it can be, right? It depends on what you're looking at. And so, and at least they're available, right? Like in some countries, like you could not do some of the research you can do in America. So I think that that's one thing. I really took it for granted that, it's super difficult to arrange the stuff that you have. But I will say that at the same time, it was never unlimited in research, because firstly, like I'm 25 years old. I'm not a professor, right? But also I felt that, it's like you have a lot of bounds to what you're trying to do. Your intentions and motivations are completely different. They're a little bit farther away from impact, in non-engineering. In engineering, like you still, like, you can build things that have a lot of impact and stuff like that. But in bio, you do a lot, you hold a lot of impact, but that's not your intention. Your intention is the information being distributed, right? So here you have limited, very limited. But in a way, it is so freeing because you can basically like choose what you're focusing on and actually like, for a fact that we're focusing on, it's like it gives back to the economy, but most importantly, it saves lives, right? Meanwhile, if you're in a university and if I discover a novel compound, then getting that into a startup, like that's the IP of the university. And then so on and so forth. It's not as, it can't be as quick to do things. Yeah. No, so I guess what I'm trying to say is that with unlimited funding, it's not truly unlimited to one mission, right? It's like you're in a lab and you're trying to basically like publish a lot of papers, right? But it's more of like a curiosity or almost like knowledge thing because of academia. Here, you're solving a problem. It's a neat balance between engineering and biology. You're solving a problem that's very specific because you don't have unlimited funding. You have limited resources. So you have to optimize your resources for a specific disease, right? Or a specific it's like instead of one semester project, it's like a lifelong project.
I see. So you're saying if you had unlimited funding, you could target all, but since you have limited funding, you cannot do everything, but you can do something.
Yes.
That's the whole point. Editing genes in a human body sounds so crazy. Is it like playing God, becoming God, and like making a superhuman? If a baby is born, you edit the genes, make it a superhuman. Is that what your vision is?
I don't want to edit any genes. I don't think that that's what we're trying to do. We're trying to solve disease. And really, like, above anything, improve the pipeline to a point where we can, when I started this company, I kind of started it alone and I was like, what am I doing? And that part I can even, tackle, like, sort of answer in a little more detail because it was very difficult, being a founder of a company that's trying to cure cancer, while like not even having, at the age of 24, and sounding crazy to everyone around me. My parents refused to help with this. And I had to somehow figure out how to get ourselves incorporated and everything. And so I think that one of the things that I realized was that if we don't do this, then people will wrongly believe that we have to wait till we do gene editing. And that's good for some diseases is never going to happen because it's not related to genes. Instead, we have really effective ways of dealing with all sorts of diseases, including diabetes, now with Ozempic. It's not CRISPR, so to speak, right? Mm-hmm. And as a result of that, I know that there's a method to the madness, right? And so really like the method to the madness is something that you can only measure through the right measurements. So our aim is not just to, a lot of people, what they do in this industry is they just like automate a lot of stuff, right? Like over and over and over again, and think that if we have enough data, we'll feed it to the ML and then that will figure out the logic. No. It's the opposite. You give the right logic, right? Because this is a machine learning, it's math, right? You give the right logic as a biologist, and then you're able to get more drugs approved, right?
So more trial and testing, when you have more data, more simulations, which we are going through, more data, which is in chemistry and physics, bringing that, more experimentation, more hit and trial, and more drugs.
Yeah, but I think that, you know what, that might not necessarily be it, because it's like the more you sort of like do hidden trial, you're not looking at the quality of the data. It's like, the quality of the data, it should depend on the cell that you're looking at, right? And so really, at the end of the day, we need more logic to this. So that's the platform, right? The platform is, we aren't just we're using our modality to figure out what we're going to test next, instead of like, oh, we're all going to test everything the same exact way. No. If you see a certain interaction in the drug cell interaction, then we're going to test accordingly, and so on and so forth. And we're going to start with drugs that have already failed, drugs that have already succeeded. And we're very early into the journey, but with that, we can not just create drugs, but also create a platform that is ML assisted using our computation, and that can be used by other companies.
So can there be a possibility when a superhuman, like if we are thinking of editing genes, can we like create next Ronaldo by splitting genes when a human is born? Next Messi, next Virat Kohli?
No.
No.
That's my answer. I refuse to believe so. There might be certain compounds that might increase your amount of muscle composition and this and that. And there's a lot of ways you can use mRNA therapy. And I think Brian Johnson has tried a couple of these, and like, that's awesome. But you're not going to do it by editing your genes. And I don't think you should, because when you edit your genes that early, you're editing all your cells, like all of them. Why are you doing that? You want to go to specific parts of your body,
which have problems, which have cancer, the risk reward is way higher.
Yes. And also sometimes you can induce cancer as a result of editing the wrong gene.
Oh my God. Yeah.
So that's the biggest problem. It's like your body's always trying to avoid this thing that you might be. Yeah. So instead, if you go about it epigenetic, hopefully in 10 years, I told you the vision is going to include like, diagnostics and so on and so forth. In 20 years, the vision hopefully also includes some more set of CRISPR-like therapies, but epigenetic, right? Maybe that's not as a result of this org. Maybe it's a result of us helping such an org. But if you can measure it, then you can actually improve it. Mm-hmm. And so the cool thing about epigenetics is it's reversible. Unlike with CRISPR, with mRNA, it's transient. You do it once, it's going to stay for a little bit, then it's going to disappear. With CRISPR, it's permanent, and it could even affect your children, which is not good. And then with epigenetics, it is reversible. And it's not transient. It stays in the next, offspring cells.
How do you see the future of Precigenetics in the next 5 to 10 years? So what will be 20 years different from now?
Okay, so the problem with CRISPR is that with CRISPR, you kind of are editing your genes entirely, right? And that's a serious problem because, you're kind of permanently affecting your genes, right? And you don't know how to target this and so on and so forth. Now, the problem is if the disease isn't even happening because of a mutation, right? It's not happening because of a change in the DNA. And then what are you editing, right? The problem's not at the that level.
So the problem is why you're editing. Figuring that why with the journey of the cell, you figure that out, and there would be better solutions in the next 5, 10 years, 20 years, and the gene editing will be done more accurately. Now, this was the entire technicalities. Now, how do you see as a business? Like, for example, Ozempic is a $1 trillion shot. It's considered the $1 trillion opportunity. A lot of people are using it and like reducing their weight and for weight loss. Do you see yourself as a $1 trillion opportunity too? What do you see yourself as a business?
We see ourselves as the platform that will enable way more such opportunities. So I think it's like a layer above that, and that's why we're creating hardware, right? If we didn't believe that, we wouldn't be here. I think Ozempic is great, but do you know how many years they waited to like get basically finally be able to make a GLP-1 agonist? That's what Ozempic is, a GLP-1 agonist. They waited like decades, right? They've been trying for decades. And now we have more compute, we have more abilities, and we have to enable better measurements to make them happen.
100%.
Yeah.
Awesome. And my last question would be here for people pursuing biology. Like they want to, I want them to just not think around B2B SaaS. People interested in biology and computer science together right now, it's an interdisciplinary field. What would you tell them? Is there a scope in job plus as a founder?
The reason why I really wanted to start my account and what I actually do with my account when I get the time is I make really long threads where I've had millions of people who've like read these threads about biological phenomena to finally be able to explain biology to engineers. And why is that? It's because people with an engineering bent of mind are the most needed in biology, but they get scared of it. That's a big part of, so for instance, after I started this company, right, I was kind of doing it alone for quite a long time. And like, Ahmed was one of the people, like maybe 2.5 months into the company, who had emailed me, and he did not have a biology background, but he said he wanted to, join the company because he's always wanted to make a certain kind of impact. And I let him know that, it's a new company. But then I was like, you know what? Like, that's the right person for this company. It's not just all the biologists, like as it's always been, because biology is a translational field. So what I'm trying to say here is that whatever you're doing, you don't need to like study biology in depth, right? For you to have the most impact in biology. I would argue that if you study something outside of biology, especially in engineering, you might be able to have even more impact on the field. The only reason why I was allowed to research in the cutting edge and medicine is because I knew how to code. And that's why an undergrad was allowed to work in such a lab, right? So I think that if you are studying whatever you're studying, you can apply it to biology, you can apply it to healthcare. And the point that I want to make to you is just be curious, because trust me, the golden age of biology is not here yet. So.
Wow. So we haven't hit the peak. We need more and more technical people, coders who understand code and can apply that computational knowledge in biology. And there are more jobs, as you see as well, or you just see more founders?
I think that a big part of why I wanted to start this company was also because I had a very lucrative, like, bunch of job offers as in computational biology that I could eventually pursue.
Like,
I don't want to.
Okay.
Yeah. But it was. But so.
But B2B SaaS.
No, but no, I had some pretty lucrative offers that were very, interesting. But I realized that, you know what, the tech companies are the only ones that are being able to, respect the biologists, right? And so much, like, there's so many other kinds of companies that are not seeing the value of the scientists. People think that being a doctor, a medical doctor, is like a better thing than being a biologist, which is my parents, since I was young, told me that cannot be true because they help people one at a time. Biologists help people as a society, right? So I think that a big part of starting this company was I wanted to be scientist-led, engineer-led. That's why, like, you know, everyone that's joins, like, it's like few people, right? Very, like, kind of like the Netflix, you know how Netflix not, so it's always few people. I've kept it to very few people, but each one of them perfect for their role. Like Emma's perfect for CPO, Max, perfect for Chief Business Officer, Chief Tech. We have Chief Science. Like we have a lot of really, really good people, and we want to keep it that way. We want people who respect science, who respect biology, and that even in tech, people did not used to have the most amazing jobs. And then the tech wave happened, right? And then suddenly, instead of being an investment banker, you wanted to be a software engineer. And how quickly did that happen? How quickly did the demand in software engineering grow, right? So in a similar way, I think with biotech, with the help of tech, but also hard tech and physics, which is what we want to bring in, we will be able to have such jobs.
So thank you so much, Ahmed and Parmita. It was a pleasure talking to you. And I hope this gives a great roadmap for anyone who want to pursue anything related to computation and biology.